Okay, let's unpack this. We're diving into the incredible electrical and chemical signals that make your brain tick, looking at some really interesting sources you've shared.
Yeah, absolutely.
Our mission today, really, is to understand how neurons talk to each other, the language of the nervous system. We'll be focusing on action potentials and a key player called GABA.
Right. It's all about those tiny electrical shifts and the chemical messengers that bridge the gaps. You've got the fast electrical stuff within the neuron and then the chemical handoff between them.
So let's start with that electrical spark, the action potential. Your sources kick off with the neuron just sitting there at rest. It's got this electrical difference across its membrane, right? The membrane potential.
Exactly. Usually around what? Negative 70 millivolts. So more negative inside than outside.
And that negativity comes from a specific ion mix.
Primarily, yeah. You've got loads of sodium ions Na plus outside and more potassium ions, K plus A, inside. Plus some negatively charged proteins hanging out inside too.
Ah, and the sources mention that sodium-potassium pump working away in the background.
Constantly. It's pumping 3 sodiums out for every 2 potassiums it brings in. Really maintains that tension, that potential difference. It's crucial.
Okay, so that's the setup. How does the actual signal, the action potential, get triggered?
Well, a signal comes in, maybe from another neuron, and it causes a little nudge in that membrane voltage, a graded potential. It's small, localized.
Kind of like a ripple.
Sort of, yeah, and it fades. But if enough of these ripples arrive and push the voltage up to a critical point, the threshold, usually around negative 55 millivolts, boom, that's the trigger for the full action potential. It fires.
And your source's stress, it's not like water flowing down a pipe, but more like a wave.
Exactly. Think of it like dominoes falling,
Yeah.
a wave of electrical change moving down the neuron's axon.
Mm-hmm.
Once threshold is hit, these special gates, voltage-gated channels, pop open.
Okay.
First, the sodium channels open super fast.
Yeah.
Sodium ions rush in, driven by both a concentration difference and that negative charge inside.
And that flips the charge.
Massively. The inside shoots up from negative to positive, peaking around plus 30 millivolts. That's depolarization. It's incredibly quick.
Wow, that's a huge swing. So how does it reset itself so quickly?
Right. At that peak, the sodium channels slam shut and become inactive for a bit.
Yeah.
Then, slightly slower, the voltage-gated potassium channels open fully.
Okay, so potassium's turn.
Yep. Potassium is concentrated inside, and now the inside is positive, so it rushes out of the cell. As those positive charges leave, the membrane potential drops back down, becoming negative again. That's repolarization.
Back towards the resting state.
Back towards rest. And your sources also give a nod to calcium ions, K2 plus Daki. They mention that calcium entering right at the very end of the neuron when the action potential arrives is key for the next step.
Which is the chemical handoff, right?
Right.
Because neurons don't actually touch.
Precisely. They communicate across tiny gaps called synapses.
So the electrical wave gets to the end.
That calcium influx triggers little sacs, vesicles, packed with chemical messengers, neurotransmitters, to merge with the membrane and release their contents into that gap, the synaptic cleft.
And these neurotransmitters float across.
They diffuse across that tiny space and bind to specific receptors on the next cell, another neuron, or maybe a muscle cell.
And that binding influences the next cell's electrical state, which brings us to GABA. Your sources highlighted this one.
Yeah, GABA, gamma-aminobutyric acid. It's the main inhibitory neurotransmitter in the central nervous system. Its job is essentially to calm things down.
So the opposite of excitatory neurotransmitters like glutamate, which you mentioned, hits the gas.
Exactly. GABA hits the brakes. It makes the next neuron less likely to fire its own action potential.
How does it do that? By making the inside more negative.
Typically, yes. When GABA binds its receptor, it usually opens channels for negative chloride ions to enter the cell or positive potassium ions to leave. Either way, it pushes the membrane potential even lower, more negative than resting hyperpolarization.
So it pulls it further away from that negative 55 millivolt threshold, makes it harder to fire.
You got it. That's called an inhibitory postsynaptic potential, or IPSP.
And GABA's doing this job in some key brain areas. Your sources mention places like the hippocampus and limbic system.
He did, which really shows how vital this braking system is for higher functions like memory, learning, and managing our emotions. It's not just simple reflexes, it's about fine-tuning complex processes.
Which makes sense why problems with GABA are linked to various conditions.
Absolutely. The sources connect low GABA, or problems with GABA signaling, to things like anxiety, epilepsy, even schizophrenia and autism. That piece on epilepsy specifically mentioned how losing GABA inhibition can lead to the runaway excitation that causes seizures.
The brakes fail, basically.
Pretty much, the system needs that balance.
And finally, there was mention of substances interacting with GABA drugs and things.
Right. Some drugs, GABA agonists like benzodiazepines used for anxiety, seizures essentially boost GABA's braking effect.
Right.
But the sources rightly flag the risks, like dependence.
Mhm.
And conversely, GABA antagonists, which block GABA, are mostly research tools because they tend to cause convulsions by removing the brakes entirely.
So putting it all together, this back and forth between the electrical action potential wave and these chemical signals like GABA applying the brakes and glutamate hitting the accelerator, that's the core communication.
That's the fundamental language, yeah. This constant dynamic interplay between electrical charge and chemical messages, maintaining that delicate balance between excitation and inhibition. It underlies everything your nervous system does.
It's just incredible to think that whole complex dance is happening billions of times a second inside your head right now, just allowing you to process this conversation.
It really is mind-boggling when you zoom in on it.
So thinking about all this intricate electrical and chemical chatter, what stands out most to you about this conversation happening inside your own brain at this very moment?